Okay, thanks. I'd like to thank the organizers for inviting me to participate in today's this year's session. And actually, I ran into John McCafferty in the hallway just a little bit earlier, and he was reminiscing about how he thought one of the most memorable talks I gave at an antibody engineering meeting many years ago was one in which, it was down at the Hotel del Coronado, and it was the one in which I was the only person to give a talk using PowerPoint. Everybody else was still using slides. And so, and I remember at the time I had help from my millennial daughter, who at the time was 9 years old, and she's now 29. So it's been a while. And I think we've made progress, or maybe we haven't, because I'm still here kind of doing the same thing. So I'm going to leave it up to you whether you think we're actually making progress. Anyway, I'm going to start. I'd like to, first of all, in addition to thanking the organizers, I'd like to thank Joe Gray for a wonderful introduction to the potential of imaging. And just for starters, I'll tell you, we're at the far right of his slides in the whole body anatomical imaging spectrum of things. But hopefully I'll be able to show you that we've made progress. Now I have to figure out which remote to use. Try this one. Okay, first the obligatory disclosures. I'm a founder and board member and consultant to ImagineAb and also have some other consulting gigs. And a brief overview of what I'd like to talk to you about. As you can tell from the title, I want to at least sort of put some context into the need for molecular diagnostics as we move into that much heralded field of precision medicine targeted therapeutics. And then I want to talk about the potential of using antibodies, not just as therapeutics, but for imaging agents, and not just in vitro settings, but in vivo as well. I'll give you some examples from our work, which up till the past few years has highly been focused in oncology. But what we're really excited about is that over the last few years, we've also made a shift into looking at potential immunological applications of antibody-based imaging. And then finally, just a couple of words on clinical translation, which is, of course, our ultimate goal. So just to remind you, precision medicine is here. So this is something I pulled off of the web. The top 10 oncology drugs by revenues in 2014. And what you can see is that 7 of them are molecularly targeted, and I think actually we have made progress because 4 of those, including the top 3, are antibody-based therapeutics. And I think it's an especially exciting time now because there's new antibodies and drugs being approved really on a very frequent basis. I think there were 2 or 3 just the past month or so, and especially in the field of immunotherapy of cancer, the checkpoint inhibitors. This has just been a really, really exciting field where we finally are starting to understand how to modify the immune system and are coming up with effective therapeutics. In fact, I read in the paper yesterday, didn't Jimmy Carter get Pembro as part of his treatment for melanoma? And he's doing really well. So this is all great. Okay, but hand in hand with very highly targeted therapeutics, we've got to understand the disease in the specific patient in front of us. So we've got to have equally detailed and powerful molecular diagnostics to go along with our therapeutics. So here's kind of where we stand in terms of molecular diagnostics in oncology. Most of the analysis we do is in vitro and laboratory-based, based on biopsies and blood tests, as well as there's some emerging technologies. Oh, move back. Okay. I'll move the microphone back. Is that better? Can you all still hear me? All right. Actually, maybe I'll stand here where I can see my slides down here. Okay, so the advantages of the biopsies are that, in a biopsy you can look at tissue and cellular composition and architecture and perhaps do some of the wonderful analyses that Joe just showed us. And we can also evaluate hundreds or thousands of markers at the same time. We can look at genomics, transcriptomics, proteins, phosphorylation, et cetera. But there are disadvantages, and I think, of course, one of the biggest disadvantages of biopsies is sampling limitations. Typically, we'll do extensive biopsies and analysis at the time of diagnosis, but when disease recurs or if there's widespread metastatic disease, it's not routine care to go in and do biopsies on those sites. Rather, we treat empirically based on what we know from the literature and just from clinical experience. And I think also this was kind of a revelation to me when I moved from doing, biodistribution studies where we looked at mouse parts in tubes to doing whole body imaging in living animals. In biopsies, you're looking at dead tissue. And what about what's going on, in vivo with living functional tissue, you lose a lot of that information when you take the tissue out. It goes hypoxic, and you fix it, you embed it, you lose a lot of information. We are fortunate that many, many good biomarkers do survive that treatment, but we do lose a lot. And blood tests, similarly, we can do very complex analyses, and we can follow over time changes, pre and post treatment, et cetera. But the disadvantages, again, we're limited to markers that actually make it into the bloodstream. So that's a huge sampling challenge as well. We're looking at an average of what's happening around the body. We're not looking, we're losing any regional information. And there is a lot of excitement, and you'll hear about some of these new approaches throughout the meeting in terms of circulating tumor cells or next-gen sequencing, et cetera. They have advantages, but they also have limitations. So I'd like to introduce the concept of in vivo molecular imaging, not just anatomic imaging, CT, MRI, but also using molecularly targeted imaging and using positron emission tomography. We use PET because it is PET and optical are amongst the most sensitive imaging methodologies that you can use. They can detect signals at nanomolar concentrations and below. And when you're talking about understanding detailed biology, those are the levels at which we need to be able to detect our targets of interest. And in terms of doing molecular imaging, we're looking in vivo. We get to look at living tissue in the context of the whole organism. Now we can have spatial assessment. We can look at regional or whole body assessment. And again, coming back to a key point that Joe mentioned earlier, we can look at heterogeneity, particularly on the whole body level. I think this actually is one of the most frightening things about cancer diagnostics and therapeutics these days is the heterogeneity that emerges. How are we going to follow it? How are we going to understand it? How are we going to know if our treatments are working? We can also do temporal assessment by doing whole body in vivo imaging. We can look at disease progression, and we can look at the same tissue pre and post treatment. PET is also quantitative, so we can measure. We can measure concentrations, we can measure kinetics and rates even. So these are the potential advantages of trying to do this in vivo. And in particular, just want to remind you that PET, it's a radioactive detection method using positron emitters. It gives you high sensitivity, resolution, and the potential for quantitation. Broad clinical availability based on this one probe shown here at the bottom, fluorodeoxyglucose. When people say PET scans these days, they mean a fluorodeoxyglucose scan in general. Wonderful all-purpose tracer for metabolic activity. And for example, you can see in this PET scan, you can see the brain, of course, is very glycolytic. Here's the bladder excretion. You can see the heart, and this. And you can also see some tumor lesions that have undergone the Warburg effect. They've transitioned to using glycolysis as their main source of energy. So because of the utility of this probe, we have PET scanners, basically in every cancer center. So the infrastructure is in place to do this scanning, but we do have only this one tracer. And one other limitation, which I'll get to, is that fluorine-18, which is the radionuclide that's used with glucose, is ideally suited for small molecule probes because it has a physical half-life of 109 minutes, which is very compatible to the biological half-lives of small molecules. But it becomes more of a challenge if you if you want to start imaging with biologicals such as antibodies. So, and the other challenge I think with FDG PET, even though it's so useful in oncology, is that it's not perfect. There are gaps. There are indolent and slow-growing tumors, such as indolent lymphomas or prostate cancers, which tend to be more lipogenic rather than glycolytic, and also the challenge of false positives, because one of the main reasons you'll get a false positive in an FDG PET scan is infection or inflammation. So we need complementary and more specific tracers, and I think that's where my group has always been interested in using antibodies exactly for that reason. We know how specific and versatile they are in terms of laboratory reagents. We know they've given rise to, actually let me just go, it doesn't matter. Given rise to, just an accelerating rate of approved FDA-approved drugs, as you can see here. And the question in my mind has always been, can we do antibody-based molecular imaging in vivo? Can we essentially do immunohistochemistry in vivo? Can we apply the exquisite specificity of antibodies to the detection and characterization of targets in vivo, and thus have a way of imaging based on the cell surface phenotype? So if you'll take a closer look at this chart, which actually been updating steadily since Henny Hogenboom published it in 1999. I color-coded the products. You can see in black there are therapeutically active antibodies. In red are the imaging agents. In purple are radioimmunotherapy products. Blue are the antibody-drug conjugates. And now we have a bispecific in the teal color. And you'll see here in the 1990s, some of the first antibody products approved by the FDA were imaging agents. It seemed to be such a straightforward and sensible thing to do. Just if you have a cancer-targeting antibody, put a radioisotope on, put it in a patient, and see where the disease is. And so 4 products were approved in the U.S. initially. Only 1 is still on the market. They have failed to succeed for a number of reasons, including poor choice of target. For example, prostacin, which is still on the market, recognizes the cytoplasmic domain of its target, so it's useful for looking at necrotic tissue or necrosis, but not so much for small viable lesions. Mouse antibodies, they were first-generation products, so most of them were based on murine antibodies. And also the challenges of working with intact antibodies. We have circulating half-lives of 5 to 7 days. And this means you have a lot of radioactivity in the background that you have to wait for it to clear before you can acquire your image. So, is it time to revisit antibodies for imaging? I think it is, due to a number of reasons. I think one is the major advances that we've had over the past 10, 20 years in terms of understanding the biology of disease and potential targets. We do have validated antibodies, as shown by the Therapeutics, so we have some good candidates to work with. And as I mentioned, also in parallel, the advances in clinical imaging, including positron emission tomography and its wide availability. Advances in antibody engineering, all of you here are part of that in terms of what we can do for optimizing these proteins. And I think the key is that we still have major unmet needs in diagnosis. I've already talked to you about the limitations of in vitro diagnosis, sampling errors, dead cells, and I think actually was at the end of Joe's talk, people alluded to the need to know the normal tissue distribution of your novel therapeutics, whether it be an antibody-drug conjugate or a nanoparticle. We need to understand where these things are going. And finally, again, hitting the nail on the head over and over again, heterogeneity of disease. We really do need to be able to understand this at many levels, and I think that whole body imaging is going to provide an answer. So let's put antibodies together with PET. What are the major challenges? This is where I started. We have an irresistible force, which is the physical half-life of fluorine-18 of about 2 hours. So here's the F18 decay curve. And then we have an immovable object, which is an intact antibody, which is going to stick around for a couple of weeks. Well, by the time the antibody gets where it's going, your radioactivity is going to be long gone. So what do? We in the antibody conjugate field have to address, we're able to, fortunately, address a lot of aspects of this potential imaging agent, and this is just a summary of the kinds of things that we have to work with, very analogous to those of you who are doing drug conjugates or toxin conjugates or other types of immunoconjugates. We have the antibody moiety, and there's a lot that we can do on the antibody engineering side to manipulate the size of pharmacokinetics, the clearance of these agents. On the right-hand side, for imaging purposes, we have a variety of different signaling moieties that we can use. I'm going to focus on the radioactive moieties, but Joe has already mentioned, for example, quantum dot conjugates, nanoparticles, fluorescent tags as well. And in the middle, we also have then defined chemistries that work for us that have either there's a great, either have the appropriate stability or lability, depending on what your final application is, and then, of course, a great move towards more site-specific and more defined conjugation approaches so that we have more uniform products for future development. So, what we've done in order to enable the combination of antibodies with PET has been to work on a couple of fronts, and one of them, of course, is looking at the radioisotopes that we're using. We and others have moved not beyond fluorine-18 and have pushed on into positron-emitting radionuclides that have longer half-lives that are much more compatible with the kinetics, the biological kinetics of antibodies and other such agents. And then on the antibody side, those of you who have followed me the past umpteen years know that what we focused on then is engineering these antibodies so they have properties optimal for imaging purposes. So of course, everything we're doing now is humanized or fully human to reduce immunogenicity and to enable clinical translation. We have focused primarily on half-life reduction, accelerated clearance. Let's get that radioactive imaging agent out of the circulation in a timely fashion so that we can image without having to wait a week. In the process, we also want agents that are inert. So we've removed the CH2 domain or the full FC when we make engineered antibody fragments. We don't want any effector functions. We just want an inert carrier for whatever signaling moiety we're bringing. We could also steer the clearance to the liver or the kidney by the molecular weight of the agent we select, and that can be important to be able to control depending on whether you need to image in the liver, for example, if you're looking for metastases from colon or pancreatic cancer, or if you need to image in the pelvic region, for example, for prostate cancer. We can select the format of the agent to facilitate a clear field of view where we're interested. And finally, as others have, we've been investing in site-specific conjugation of our imaging tags. And this is just a brief review of what we have accomplished over the past years, which is that we focused on engineered antibody fragments that maintain bivalency for the target of interest, and yet have much more rapid clearance properties. And first of all, you'll notice that the Diabody and the mini-body are the fragments that we have focused on. The Diabody is below the threshold for first pass renal clearance, so it clears very quickly, as shown here. The mini-body is above the threshold for first pass renal clearance, so it clears via the liver, although since it lacks the CH2 region and it lacks interactions with the FcRn receptor, it's still fairly rapid clearance and it disappears from the circulation within 24 hours. So these two are the mainstays of what we've been working on. I think there's just a summary there, but in short, we are able to get high tumor-to-blood ratios using these engineered antibody fragments, but at much earlier times than you can with an intact antibody. And that's shown, of course, pictures with 1,000 words. Here are a series of cognate anti-prostate stem cell antigen fragments, all labeled with I-124, looking in the same tumor model. You can see excellent targeting to the positive xenograft on the right side of each of these animals on the right shoulder. You also see some bladder activity with these smaller fragments. But what you will see is that we're able to, with the fragments, generate the contrast in the images either the next day with the mini-body or the same day with the Diabody format compared to an intact antibody where we have to wait a week, and you can still see there's some background activity in the mouse. We still have to wait a week or so for all that background activity to clear. Okay, so, and just to focus a little more on the Diabody for same-day imaging, I think importantly you'll see that with these very small fragments, we get peak uptake in 1 to 2 hours after injection with a half-life of 3 to 4 hours. So those are kinetics that are fast enough for labeling with fluorine-18 for rapid imaging. And we actually, over the years, have demonstrated this in a variety of animal models. At least here is an anti-CEA or anti-HER2 or anti-PSCA Diabodies labeled with fluorine-18, and you can see a localization to the antigen-expressing xenografts, and these are all within 4 hours of injection. Okay, and then again, continuing on the theme of doing site-specific conjugation, we've developed cysteine-modified Diabodies. If you take the Cys-diabody and just conjugated at random so that you can chelate a metal. So this is just DOTA, a chelating agent using NHS chemistry. You'll get a mixture of fragments at the end with different conjugation ratios, 4, 5, 6, 7, or 8 DOTA moieties per Diabody. Or you could take that same protein, lightly reduce with TCEP, now use maleimide chemistry that's thiol-specific, and instead what you find is you can just modify that single cysteine residue that's on the C-terminus of the protein. So this really gives us a powerful way to have control over the location and the stoichiometry of the modification, and it gives us a much more uniform product. And these Cys-diabodies actually can carry a variety of cargoes. So in the lab, for example, we put low molecular weight fluorescent dyes on, or site-specific radio labeling. Almost all of our radio labeling is done now in a site-specific fashion. We have put these small proteins onto nanoparticles such as quantum dots, and as Joe mentioned earlier, this may be advantageous because these Diabodies are only about 5 by 7 nanometers as opposed to intact antibodies, which are more like 15 nanometers. You can do site-specific fluorine-18 labeling. And this is just one quick example of a fluorescently labeled Diabody, which we're using in an intraoperative setting, looking at prostate cancer. These mice were injected intramuscularly with the tumor, and it was allowed to grow, and then systemically administered a Cy5 labeled Cys-diabody, and in the top you can see just opening up the mouse, then the surgical fellow removed most of the tumor and left this little bit here, but what you can see under fluorescent light is here's the bit he intentionally left, but here's a little bit extra of margin where he did not under visible light see that there was still tumor there. So this is an example of what we could do. And for further examples of what you can do with this approach, student Wen Ting in the lab has a poster, not a Cys-diabody, but rather a Cys-minibody, which he has dual labeled with I-124 and Cy5, and is now imaging in mouse models of prostate cancer. And so basically, this is a platform approach. You can take any antibody with the specificity of interest, reformat it into one of these fragments. To a first approximation, you'll get much more rapid clearance kinetics. You can use very standard chemistries now to attach PET radionuclide and have yourself a PET imaging agent that you can use either for same-day or next-day imaging. And I think importantly in oncology, there are a lot of questions you can start to ask because it's a really general approach for imaging based on cell surface phenotype. Beyond just asking, is there tumor present? You can start, depending on which antibody-antigen system you're using, you can gain information on tissue of origin, activation markers, elevated expression of receptors. If those receptors are targets for targeted therapy, you can use that for therapy selection potentially. You can also look at occupancy. Is your therapeutic actually getting to those sites, and is it occupying those sites? To what extent? We can look at things such as internalization and catabolism in vivo. These are important questions, for example, if you're working on an antibody-drug conjugate. We start to confirm mechanism, either direct mechanisms or indirect mechanisms, response to therapy, and as I mentioned earlier, there's always the potential for quantitation of these characteristics. So I want to just give you a couple of quick examples, recent examples of what we've been able to do in mouse models. So just coming back to the minibody for same-day or next-day imaging, I want to give you two examples of either detection of small bone lesions or response to therapy or a resistance to a potential application in looking at resistance to therapy. First of all, this is the prostate stem cell antigen antibody that I introduced briefly earlier. It's a marker that is expressed at low levels in normal tissue, including normal prostate, lumen of the stomach, and transitional epithelium of the bladder, but upregulated in prostate, pancreatic, and bladder cancer. We went through the process of humanizing, affinity maturing with Jim Marks's help, reformatting into a mini-body, radio labeling, and have done now a very careful series of quantitative ImmunoPET studies comparing I-124 and zirconium-89, which are PET radionuclides with longer half-lives. And then in terms of extending the application, here's an example where we were looking at intratibial, very small intratibial tumors at 4, 6, or 8 weeks after injection. This is just a set of 4-week images. And the top row shows a 6 mice scanned with a sodium fluoride bone scan. It's a very busy scan because these are young mice, they're growing, they're very active, and any place there's bone growth or remodeling, you'll see signal. And what you're looking for, I don't know if you can see it with the lights, you're looking for asymmetry below the knees in these mice. So here in this mouse and maybe this mouse, you can see a little tail of activity on one side, and that's due to the tumor, the intratibial tumor growing in those mice. But if you, instead of using a generic, bone remodeling agent such as sodium fluoride, you use the very targeted tumor-specific PSCA minibody with I-124, the bottom set of scans, at 4 weeks you can see these very tiny lesions inside the tibia, and then by 6 weeks, 6 out of 6 mice, you can see those lesions. So this shows that you can detect very specifically, here's the quantitation, very specifically the presence of these very small tumors using an ImmunoPET approach. And then the next example is a response to therapy example where we're looking at the fact that following anti-androgen treatment, PSCA is an androgen-dependent marker, and after treatment, what you can see is here the vehicle-treated mice on the top. Here are the mice that have been treated with enzalutamide in the bottom row, and what you can do is actually quantitatively measure that there has been downregulation of PSCA expression, either in the presence or absence of, looking at whether the tumor shrinks or not. So we're able to pull apart those two aspects of response. Does the tumor shrink, but has that target also been downregulated, which validates the mechanism of action of that agent? And then this final example comes back to a different target, c-MET, which and we heard a little bit about c-MET earlier, and this was a project that we did in collaboration with Parag Malik and Sam Gambhir up at Stanford, who were looking at resistance to targeted treatments using EGF receptor inhibitors, either antibody or small molecule. One of the mechanisms of resistance to EGFR therapies can be upregulation, overexpression of the MET cell surface receptor. And the question was, is this something that we can assess non-invasively? So what we did is we went through a phage display library to identify novel antibodies that recognize these are fully human antibodies that recognize MET. And this is just some of the characterization to show that we actually developed quite a few promising antibodies with nanomolar affinity for MET. And then when we took one of these and looked at a model of the HCC 827 non-small cell lung cancer, plus or minus a gefitinib-resistant subline that was developed by Jeffrey Engelman's group, that resistant cell line shows fourfold overexpression of MET. And what Kyu Lee did, he's a past graduate student in the laboratory, he made an anti-MET mini-body, radio-labeled it with zirconium-89 for PET imaging, and here you can see in the scan, here's the resistant cell line versus the parental cell line, and you can clearly see a much stronger signal. We're picking up that difference in terms of overexpression of this c-MET receptor on the cell surface. In culture, that represents 260,000 copies per cell versus 40,000 copies per cell in the parental cell line. Okay, so that all looks promising in oncology, but what I'd like to do is spend the last section of this talk introducing you to what we've been doing that is, I think, much newer and equally exciting and compelling, we hope, which is to move beyond oncology, to move beyond simply looking at cell surface targets on the cancer cells themselves, but to start to look at, a critical component of the microenvironment, the immune cells. Actually, it's a critical component of the entire body. So as I mentioned earlier, FDG-PET is nonspecific when it comes to tumor cells versus immune responses. But we also have this opportunity in that there are all of these CD antigens, the clusters of differentiation antigens. I went through, 499 of them, or how many there are now, but I'm sure you're well familiar with many of them. Great markers, very well-defined targets and markers of lineage, differentiation, cell activation state, and a corresponding very well-defined set of antibodies that recognize these markers, although we can argue about how well-defined they are in the session this afternoon that Andrew's leading. Importantly, I think there are tremendous potential applications for imaging immune cell subsets in immune cell responses and inflammation in general, and in particular now in the field of cancer immunotherapy. So, and this work was inspired by Tony Ribas, one of the collaborators shown here on the bottom. We've done all of this work in collaboration with fellow faculty at UCLA, including Owen Whitty, Tony Ribas, and John Timmerman. Tony treats melanoma, and he actually did a lot of the work with the checkpoint inhibitors in melanoma. And one of his big challenges that he brought to us some 10 years ago was that it was really hard to figure out what was going on in these patients that he was treating with the checkpoint inhibitors because their FDG signals, the FDG PET, would still be strong after treatment, and they did not have any way of understanding whether that signal was either due to the tumor cells continuing to grow or an active immune response against the tumor cells. They're both highly glycolytic processes. So he got us interested in the concept of imaging the immune cells specifically, as distinct from the tumor cells, using cell surface antibodies. And this really is central in the field of cancer immunotherapy. I give some examples here of where there's been great progress in the field, not just the checkpoint inhibitors, but the chimeric antigen receptor, modified T cells, vaccines, immunocytokines. We had a whole fireside a couple of years, big keynote session, was it last year, on immunocytokines? And as I mentioned, recent approval of bispecific antibodies, and we know a lot of groups and companies are working on bispecific antibodies. Now, in many of those cases in oncology, all of those treatments converge on the cytotoxic T cell. And Tony Ribas says it's the cytotoxic T cell that is the actual drug. We're just trying to induce it to be in the right place and be in the right activation state. So that kind of planted the idea in our minds that we really should try to find a way to image CD8 T cells. So this is work by Richard Tavares. He's sitting also over here in the audience. And this actually was a giant step backwards for my lab, because up till now we had been working exclusively human targets, human or humanized antibodies, human xenograft models, et cetera, or transgenic mice expressing the human targets. But in order to take our first foray into this field, because of the availability of mouse models and the lack of cross-reactivity of antibodies, the challenges of doing fully humanized mouse models. In our little academic lab, we decided to start with mouse systems. So Richard identified some rat anti-mouse hybridomas that we could work with, including for CD8, these two hybridomas shown here. And first, a brief bit of introduction to the CD8 target itself. CD8 in mice is an alpha-beta heterodimer, or sometimes an alpha-alpha homodimer. It's the LYT2 is the alpha chain gene, and it actually occurs in mice in two major allelic variants. The 2.2, as shown here, has a methionine at residue 78. The 2.1 has a valine at residue 78. One of the starting hybridomas is specific just for the LY2.2 allele. The other one, the 169, is pan-specific. As shown here, and Richard initially started his work on the 2.43 minibody, converted it, generated a minibody, radio-labeled initially with copper-64 for PET imaging, and here is imaging in a normal black 6 mouse. So first of all, you see clearance to the liver and a very hot signal in the liver, because not only is the minibody that's not bound to target cleared to the liver, but also copper-64 itself is scavenged by the liver. So that's non-specific uptake. But what you can see is the spleen and a very nice map of all of the normal lymph nodes in the normal mouse, including if you have really good eyes and if the lights were down, you could see even the popliteal lymph nodes, which are like a 0.5 milligram to 1 milligram in size. And important controls for specificity include blocking the mouse with excess cold minibody ahead of time, so you see all the signal disappears except for the non-specific uptake in the liver. And finally, in the last panel there, I think a really important control, which is that the parental antibodies are depleting. So Richard treated the mouse with the parental antibody ahead of time to deplete the T cell population, and then imaged the mice, and what you can see is indeed he has been successful in biologically manipulating those mice. They don't have any CD8 T cells, and you don't see the signal. So that looked encouraging, and then of course because of the strain differences, another great control was to do this in a C3H mouse, which has the other allele, and you can also see in the mouse with the other allele that you do not see the signal in the spleen or the lymph nodes. And to me that reminds me why we're all here. Because here we've taken these antibodies and we've reformatted them into minibodies, and we've radio-labeled them, and we're using them in vivo, and this antibody is still able to distinguish that single amino acid difference between those two alleles. That's why we're here. Okay, so he's gone on to apply these antibodies in a number of models, and for example, here is a model of hematopoietic stem cell transplantation, where we take one mouse, the recipient irradiate it, and then we take a donor mouse, take bone marrow cells, put them back into the recipient mouse. They're marked differently on CD45, so we can follow the reconstitution process, but I'm not going to show you all that detail. But also to mention in this case, now we've moved on, Richard has now moved on to an optimized pan-specific reagent. So this is from the other hybridoma, the 169 hybridoma. It's a Cys-diabody that's been optimized for imaging. And when you image these mice over time post-stem cell transplantation, first of all, it's a Cys-diabody, so you'll see it clears to the kidney. Sometimes it's a little hard to see the spleen. The spleen sits atop that kidney that's on the left, but I think it's the right kidney. Whatever. But you can also see, if you look at the lymph nodes over time at 2, 4, or 8 weeks, or the thymus and the spleen, you can see the repopulation of those lymphoid organs with the CD8 T cells. So, and then finally, what triggered all of this work in the first place was to be able to look at CD8 T cells in the context of tumor immunotherapy. So this is just one of the models that we've been using in collaboration with Tony Ribas. CT26 colon carcinoma, it's syngeneic to the Biopsy mouse. This is actually a fairly immunoresponsive tumor in the mouse model. These are tumors that are implanted subcutaneously in the shoulder region. And in this example I'm showing you, the mice were treated with an anti-CD137 therapeutic antibody. It's also known as 4-1BB, and this is a general immune stimulatory antibody. And what you can see here is the timeline. The tumor cells are implanted at day 0, day 7 through 13. The mouse is administered 4 doses of the anti-CD137 antibody. Day 15, Richard injects. Day 16, we image. And typically what you'll see is that over time, the untreated tumors keep growing. The tumors in the treated mice start to regress, and in fact, they're typically gone by about day 20. But we're imaging at day 15 when there's still tumors there. And importantly, what you see is if you remove those tumors from the mice and stain them for the CD8 T cells, in the untreated mice, you just see a rare CD8 T cell. In the treated mouse, by immunohistochemistry, you can see massive infusion or massive recruitment of the T cells into the tumor, and between migration and proliferation. So what happens now when we try to image this mouse with the CD8 specific T cell CD169 Cys-diabody by PET, the results are shown here. So on the left is, oh, I'm sorry, this is labeled wrong. On the left is the control. I rearranged them and wanted to put in the transverse images. On the left is the control, an untreated mouse. And what you will see here is a large diffuse signal. First of all, the tumor's large, and you do see, at least on the maximum intensity projection, a very diffuse signal. We're looking at the T cells, not the tumor. And you can also see a little bit of spleen and a little bit of the rest of the immune system. And here's the transfer section as well, which I'll come back to. This is the treated mouse. I switched these two labels. And what you can see now is the tumor, first of all, it's a lot smaller. You'll also see the signals a lot more intense. And I think the other thing that's of interest is this is a general immune stimulatory antibody, so not only do you see a very hot draining lymph node, you also see systemic activation of the entire immune system. You see the spleen is large and full of T cells, as well as all of the lymph nodes in the body. So this is something that you can, the kind of information you can get by doing whole body imaging that you cannot get from a biopsy. Finally, here's a control, which is the blocking study. So it's a treated mouse, but pre-blocked with the excess cold CD8 T cell agent. And what you can see is all of that signal disappears now, just the nonspecific uptake in the kidney. So that is confirmation that the signals that we're seeing are specifically due to the CD8 T cells in the mice. And I showed you already the previous slide, the immunohistochemistry. Interestingly, here are the cross sections, and what you can see is in the untreated mouse, most of the signals confined to the periphery. You don't see T cells inside the tumor, whereas in the treated mouse, you can see they are just wall to wall, and this kind of reflects what is seen in the immunohistochemistry. We've done quantitation to show you that here's the tumor to blood ratio in the treated mice, the blocked mice, and in the untreated mice. Likewise, you can quantitate the T cells and show that after treatment we have an increase in the T cells, and also in the biodistributions you can see under the blocking and control situations, it's just the lymphoid tissues where you see those signals go down. So if you're interested in hearing further, I recommend that you attend Richard's talk, which will be Wednesday at 11:30. We'll go into this in much greater detail and show additional immunotherapy models where he's been applying this approach. We've also then extended the work not just to CD8 T cells, but also to other immune cell subsets. Here is an example of imaging CD4 cells, similarly using a rat anti-mouse hybridoma and generating the GK1.5 Cys-diabody that's been labeled with zirconium-89, again for PET imaging. This is just the same type of control study showing the map of the mouse's immune system, immune lymphoid organs, and either in the blocked or the depleted setting, you can see that CD4 image signals are disappearing. And then on the CD20 side, this is work now of Kirsten Zetlitz, who's also sitting in the audience, who is now working, we're back into the human arena, where she's taken the GA101 obinutuzumab humanized anti-human CD20 antibody, reformatted it into mini-bodies, Cys-diabodies, and compared and evaluated formats, radio label, et cetera. This is one we're particularly interested in because now we're finally getting back to a fluorine-18 radio-labeled probe. And these are just some images that have been obtained in human CD20 transgenic mice. We wanted to be able to image what the normal immune system looked like in the transgenic system, and then later on to be able to use this mouse as the basis for tumor imaging. So what you can see, here's the classic FDG PET scan. These are just normal, non-tumor-bearing transgenic mice. You see the Halderian glands in the brain, you can see the heart, you can see the bladder, not unlike what we saw with that patient scan earlier. But now here with a fluorine-18 labeled CD20, human CD20 specific agent, now what you see instead is the spleen. You can see some lymph nodes if you zoom in carefully, and then these are some more lymph nodes in the mouse. And again, if you'd like to see further details on this work, including imaging in several tumor models, I refer you to Kirsten's poster that will be presented later on in the meeting. So, in summary, I think what I'd like to close is to show you, I've given you examples where we've developed an anti-murine CD8 Cys-diabody for ImmunoPET imaging of CD8 expressing T cells in vivo. And we've been using these to detect T cell repopulation in a mouse model of hematopoietic stem cell transplantation. I didn't go into details, but one of the things that we have worked on is the quantitation. We've had to correct for the path length of the positrons that are emitted by the zirconium and the I-124, as well as for the resolution of the scanners. There's this effect called partial volume effect that comes into play, especially when you're trying to image small objects such as lymph nodes in a mouse. And then, as I showed you at the end there, we're able to now use this approach to detect CD8 T cell infiltration in tumor immunotherapy models. We also, as I mentioned, have the CD4 specific Diabody. We also have anti-human CD20 specific imaging agents. So. In particular, in the latter case, these are agents that are going to be readily translatable to clinical use. So just a couple words on clinical translation, stepping back to the prostate stem cell antigen-specific agent I showed you earlier in the talk, where we generated a humanized, affinity-matured mini-body that was labeled I-124 in collaboration with City of Hope, and Paul Yazaki, who's sitting here in the audience, many years of collaboration, produced cGMP, the clinical-grade antibody, filed the IND, 2,000 pages worth, IRB approvals, and that agent is now in clinical evaluation at UCLA in patients with prostate cancer, pancreatic cancer, or bladder cancer. And then a more recent example, this is now work coming out of Memorial Sloan Kettering and Cornell Weill, looking at the PSMA-specific antibody, the J591. The intact antibody has been studied extensively by the groups in New York. And for example, they show this one study where PSMA expression on the surface is actually a good readout of anti-androgen therapy. I showed you earlier, PSCA depends on androgen, so when you treat with enzalutamide, the PSCA signal goes down. The opposite seems to happen in these models where you treat with the anti-androgen and the signal for PSMA goes up, so it gives you a readout of how effective you're ablating the androgen axis. So they showed that with intact antibodies as shown here, as an example of how we can use a cell surface marker to understand what's going on intracellularly. And then also Jason Lewis's lab took the anti-PSMA mini-body and Cys-diabody, labeled them with zirconium-89, and also showed what we've been discussing all along, which is excellent targeting to subcutaneous tumors that express the marker, as well as clearance of the mini-body to the liver or the Diabody to the kidney. And then more recently, led by Nita Pandit-Taskar and Mike Morris on the clinical side, studies have now been done imaging men with known metastatic prostate cancer. This is just one example where it's the same patient either imaged with the technetium-99 bone scan, which is the standard for looking at bone metastases. They also do FDG PET scan, as you can see here, and finally an image PET scan, but now using the zirconium-89 labeled anti-PSMA minibody as a tracer. And what they're finding in general is detection of significantly higher numbers of lesions using the prostate cancer-specific minibody compared to conventional imaging. So they're in the process of analyzing and writing up that work. But I think it certainly shows that these are translatable and potentially very useful tools. So to summarize, I think we can make minibodies and Cys-diabodies from any antibody of interest. Importantly, they're, the ones that we make should be non-immunogenic. They should be biologically inert. They demonstrate rapid targeting and imaging, and we can either steer the clearance to the liver or the kidney, depending on what our final application is. We can use site-specific conjugation of either the cis minibodies or the Cys-diabodies to provide a more uniform product that's going to be more translatable. And we are continuing our efforts. There's a lot of work going on in the lab, which is not quite ready to present yet, on site-specific labeling using fluorine-18 for same-day imaging. And in oncology, we can use these as in vivo biomarkers to profile tumor biology, to look at normal tissue expression, therapy selection, delivery, coverage, internalization, and metabolism of therapeutic antibodies, and we can look at response to therapy. And I think, as I mentioned, what we're most excited about is the ability to now look at immune cell subsets to profile immune responses in vivo, to look at the subsets, to be able to look at expansion, trafficking, activation, and in a way to be able to look at the sort of the biomarker microenvironment either in your tumor or a draining regional lymph node to be able to understand what is going on with your immunotherapy. And I think that obviously there are going to be many applications beyond oncology and cancer immunotherapy. We are interested in, we're starting to move into models of autoimmunity. There's just immune responses are so central to so many aspects of normal biology and disease. So I'd like to acknowledge the people in my lab. Richard, Kirsten, and Wenting are all here at the meeting. And this is just shows you some of the other key members of my laboratory, present and past. Scott Knowles did a lot of the PSCA work here. You did the library work and the CMET antibodies. Amanda's working on the CD4 and mouse models for looking at CD4 subsets, infiltration in disease models. And my many collaborators at UCLA who have provided either help with the, well, not, have been key collaborators in terms of providing the models that we use, as well as the instrumentation and the radiochemistry. And then also at Imaginab, Tova Olafsson, many of you may remember, was a long-standing member of my laboratory. She is now down at Imaginab. And then, of course, our collaborators at Memorial Sloan Kettering for the more recent clinical studies that I showed. So thank you.
Great talk, Anna. Do you see the, I mean, especially the T-cell stuff, do you see this as becoming a sort of an integrated part of therapy in cancer, to sort of treat with some therapeutic agent and then come in and see what the T-cells are doing afterwards? That's
absolutely the hope. I'm pretty sure that we'll be, especially for these immunotherapies, the T cell is the active agent, and we very much think that you might be able to do a protocol where you could scan and then treat and then scan afterwards and look for an increase, influx of T cells and or proliferation. I think this is going to be really important too, because tumors are heterogeneous, immune responses can be heterogeneous, and to be able to understand throughout the body, even on a lesion by lesion basis what's going on, it's going to be important to understand whether or not your therapeutic is working. So absolutely, that's where we're hoping that this will be useful.
Very nice talk, Anna. Real quick question. I mean, the immuno-oncology imaging, of course, is critically important, like you said, and I think it's very exciting. Do you have a sense of what the minimal number is, or the max of cells that you can actually image? CD137 is present on a number of, immune cell populations. Could you image Tregs, for instance? Could you image, I mean, what's the lower level?
So I think the lower level is going, in the mouse model, is going to be, Richard has done some calculations because of how many T cells are in that tiny little lymph node that we do visualize by PET, even with the partial volume corrections and all. And he estimates something like 70 to 100,000 T cells, all very concentrated in one spot. We're able to see that. We've been doing a number of studies, both in my lab, but also ImagineAb's been doing studies, and we're in the order of 100 or hundreds of thousands of cells. I think, it's a key question, and it's going to be different once we get to patients. But I think these are still going to be significant numbers. So we're working very hard on understanding. For example, people are very, very used to looking at, the microscopic level in immunohistochemistry and what cell densities look like. So we're now aligning that with what PET signal we're seeing in tissues, so that we can come back with more concrete answer to that question.
Thanks.
So can I have one question, actually? So,
okay.
All the data you provide today is mostly for the mouse models, right? But if you wanted to develop real sort of precision medicine kind of companion diagnostics, then you should apply to the patient. So what do you think is most important aspect between the mouse model? If you want to develop the patient-related sort of imaging tools, then what should be the most important aspect that we need to consider to develop the patient-related imaging tools?
Yeah, so that work is going on at ImagineAb. And so obviously you need human-specific reagents, and you need humanized or fully human reagents, and I think it's also really important. Safety is going to be an issue, so we need to both look at the biological activity of those proteins. As anybody who's doing any kind of immunological or agent, you don't want to kill the T cells or inactivate them, but you don't want to stimulate them either. This is just an imaging agent, so we have to look at the biological activity of those reagents, and then we also look at the radiation dose aspects of that as well. But that's all well understood. The radiation dosimetry, how much activity you can administer, what the dose deposition would be in different tissues. So all of those factors are important to look at, and I also think it's really important. It's really hard to extrapolate from the mouse to the human. So I think ideally we need to get into patients as soon as possible, and then just start to study these, in the context of real patients.
Hi, great talk. I was wondering whether you did imaging study and correlation between the size of the antibody and the imaging in tissues which are not that accessible, for example, bone metastasis in prostate cancer, because I'm thinking maybe you will have a different tool there. You will need bigger molecule, you need a better pharmacokinetics. Is it true?
Yeah, so there's a trade-off. On the pharmacokinetics side, there is a trade-off between exposure and uptake. You need exposure for uptake, but then we also are balancing exposure and clearance because we also want the background activity to clear. So the intact antibodies will give you higher absolute levels of uptake, and you have to wait longer. And I think those properties are ideal for therapeutics, but maybe not so much for imaging. One of the advantages of using fragments is that they theoretically, and have been shown in animal models, to penetrate better because they are a lower molecular weight, and often in tumors you're limited by diffusion, as opposed to having uniform transport and perfusion. So in that sense, you can counterbalance by the fact that smaller reagents will permeate and more homogeneously into tissues such as tumors. So I think there are just multiple factors that all of them have to be sort of optimized simultaneously to get the properties that you want. But I think that I think for the imaging purposes, I think the fragments actually hold out a lot of promise.
Okay, thanks very much, Anna. Can we give all the talkers this morning a round of applause?